So what we're going to talk about today are mucosal infections. Basically, you've got two lectures on infections, and we had to carve the world up one way or another, and we've decided to do it as mucosal infections and invasive infections. So just to remind you what we said this morning about the location of bacterial infections, that we have this, if you like, this blob with a hollow tube running through it, which is you. The lumen of your gut is part of the outside world. There's a mucosal surface there. And bacterial pathogens, to actually cause disease, have to colonise mucosal surfaces to get a foothold. And they require adhesins and other virulence factors. Now, it's an interesting point worth noting there that to actually colonise those surfaces, they actually are using the very same kinds of mechanisms as commensals do, because commensals have to stick around as well. So as I mentioned earlier about your bowels trying to, peristalsis pushing things out, if an organism wants to stick around in the bowel as a commensal or a pathogen, it has to be able to adhere to the mucosa and so on. Now, some pathogens just get into the mucosa, stick on the mucosa, maybe get into the cells of the mucosa, but they don't go anywhere else, and that's what we're going to talk about today. Sometimes they produce toxins when bedded down there, which have distant effects, but the bacteria themselves don't go anywhere else. Other pathogens can invade the tissues of the body more widely, get into the bloodstream, and disseminate it to sites distant from their point of entry. And that's the subject of the second lecture of this pair, which you'll get next week. Which you'll get on Thursday. So again, just to draw this picture of, this is the mucosa, all that green slimy stuff there is the mucus. We've got the epithelium there shown in this kind of yellowish colour, and the bacteria in blue, not to scale, I hasten to add. And these pathogens, as I say, they remain restricted to that epithelium. Sometimes they can invade the cells of that epithelium, although we're not going to talk about any such pathogens today. The key point is they don't go further. So they release toxins, other virulence factors that may damage or derange the cells, and they cause their damage locally. Sometimes the toxins can also diffuse more widely. Now, last year I put 9 different pathogens in, and I was told off for kind of cover too much. So we've cut this down dramatically to 4 different diseases and the associated pathogens. And this is illustrative of the kinds of processes that go on and the kind of context in which these infections occur. And there are, of course, many, many dozens of similar important diseases on these surfaces and in other body surfaces, and there are probably hundreds, if not thousands, that have been catalogued as occurring at one time or another. Let's start with diphtheria. What is diphtheria? This is an acute toxin-mediated disease caused by a bacterium called Helicobacter diphtheriae. It's always good if you want spelling challenges there, because there's a PH and then a TH, which is a very unusual cluster. It's a gram-positive bacterium, and it's aerobic in its growth requirements. It likes to live in air. And when we call it a bacillus, we mean it's kind of rod-shaped. Now, it's spread by droplets, and it enters the body through the respiratory route. So someone is coughing or breathing, and then you breathe in their air and you get it. A few days incubation period, and then it typically involves the pharynx and the tonsils. And it starts off as just another sore throat, but it gets worse and worse, typically, and you end up with this leathery adherent membrane. So in fact, the name diphtheria comes from the Greek word for leather. And you can see it in these pictures here at the back of the throat there. You can see this stuff stuck down, this kind of whitish greyish stuff stuck at the back there. And that membrane can actually spread and block the airflow into the lungs, and people can die from asphyxiation in the worst case. In addition to those complications that you get locally, you get distant complications. You get damage to the heart muscle, myocarditis as it's called. And in the acute phase of the illness, this can cause cardiac arrhythmias and sudden death, and it can cause prolonged damage and arrhythmia. And this picture here is actually of my great-grandmother-in-law, my wife's great-grandmother, who suffered from diphtheria shortly after that photograph was taken in the, would have been about the 1920s. And she had sustained damage to her heart, and she had a cardiac arrhythmia for the rest of her life as a result of catching diphtheria as an infant. She actually died in later life from a stroke, which was probably worsened by the cardiac arrhythmia. So in a sense, the diphtheria didn't kill her immediately, but it did contribute to her death many decades later. The other thing you get is this neuropathy, damage to the nerves. It's a so-called motor neuropathy, which means that it damages nerves which are feeding impulses to muscles to control them. You get paralysis of the soft palate, so it's very difficult to swallow things. You get then eye muscles, limbs, diaphragm, and in the affected, and in the worst case, the paralysis of the diaphragm means the person can't breathe and they will die without assisted ventilation. And that will slowly get better over time. So we mentioned this is a gram-positive aerobic bacillus, and when we look down the microscope, it's arranged in these kind of V or L shapes, or as the earlier generations of bacteriologists used to say, they thought it looked like Chinese letters. I guess someone who's familiar with the Chinese alphabet might say, well, they look like Roman letters to me, but that's what they used to say back in the day. You also can see particular granules in them with a particular stain called Albert's stain. When we grow it in the lab, we have a particular indicator medium, which is called tellurite agar, and that's because these colonies from the bacterium can reduce sodium tellurite to tellurium, and you get this black colour forming in the colonies there. The other thing that we do, certainly used to do in the old days before we had molecular methods, was to test for the toxin. And the gold standard for this was doing it in a guinea pig, where you inject an extract from the bacterium and you see if the guinea pig gets a diphtheria-like illness. Development on that was this test here, shown as the ELEC test, where instead of doing that, you actually use an antibody to the toxin, and you look for arcs of precipitation where the toxin and antitoxin meet in the agar, and you can just about see those on there around the positive control. Nowadays, we would tend to use a PCR-based test, a DNA-based test as well, certainly as a preliminary screening test. So what about the toxin? How does this disease come about? Well, it comes about because there's this toxin. Toxin gene is actually encoded by a bacteriophage. And if you remember in the lecture I gave you a few weeks ago about microbial genomes, bacteriophages, they're bacterial viruses, but they can actually settle into a bacterial genome, integrate into the genome, become what is known as prophages. And that's what happens here. The toxin gene is carried into the genome of a non-toxigenic strain of Corynebacterium diphtheriae by a toxigenic phage that becomes a prophage. Now, I mentioned in the earlier talk today about the importance of iron, that body fluids are very low in iron. And the expression of this toxin is regulated by levels of iron, but not in the positive sense, in the negative sense. So when iron levels are low, the toxin gene is switched on. And so when the Corynebacterium diphtheriae is sensing that it's in body fluids that are low in iron, it switches on the toxin. This is an AB5 toxin, as we mentioned this morning. There's this B subunit, it's got two different domains in there, and then an A subunit, which is the active part. And this is an ADP ribosal transferase. Now that's a little bit of a mouthful, but what that means is it takes an ADP ribose molecule and it sticks it on to whatever the target is. And in the case here, it's a particular residue on an elongation factor involved in protein synthesis known as diphtheria. And when that diphtheria residue gets ADP ribosylated, that then inhibits protein synthesis, and that causes the problems associated with the toxin. Diphtheria is a medical emergency. If someone has diphtheria, there is a serious risk that they may go into respiratory obstruction. So some of that membrane may just get lodged in there between their vocal cords and block their airway. They also need antiserum urgently, and we give them antibiotics as well, erythromycin. Whether antibiotics make much of a difference to outcome is unclear, but they stop the person shedding the organism fairly quickly. It's a public health emergency as well. It's what we call a notifiable disease. So that if a physician sees a case of diphtheria or a suspected case of diphtheria, they have a legal requirement to notify the public health authorities immediately. And there's a need then to go on and do contact tracing and to give prophylaxis, to give antibiotics to those who've been exposed to what we call the index case. How do we prevent this disease? Well, luckily it is a preventable disease. With routine childhood vaccination, we have seen this illness off. It is now pretty much eradicated from the United Kingdom. We only ever see diphtheria when we get it in returning travellers coming from other parts of the world bringing it in. It's no longer endemic here. Sadly, when it does come, it does sometimes spread, so you'll see a small outbreak associated with someone coming from a country where diphtheria is not well controlled, and it might sputter on. You might get 1 or 2 cases after the first case, and so on. There's a lot of lively debate also about the need for adult boosters. There have been a couple of outbreaks of diphtheria in developed countries associated with adults. So although we normally think it's a childhood disease, there was an outbreak in Sweden in the 1980s associated with adults. And so there's a lot of question about whether we should all be getting boosters every 10 years to prevent the risk. Okay, that's a quick run through of diphtheria as one example of a kind of disease of the mucosa affecting the respiratory tract. Pertussis, or whooping cough, is another such infection. This is caused by another bacterium. This one has the particular name of Bordetella pertussis. It's what we call a fastidious gram-negative aerobic rod. And by fastidious, we mean it's quite choosy about what kind of culture medium it will grow on. It won't grow on any old culture medium. You have to have a very particular, very rich culture medium for it to grow. It affects the upper respiratory tract. Airborne transmission, as with C. diphtheriae, and particularly severe in the children and in the elderly. And it gets its name from this particularly nasty cough associated with it. It's a kind of staccato cough, and it's the whooping cough. But it doesn't spread into the bloodstream, thank goodness. It just stays in the respiratory tract. There's 3 stages to the infection. You have what we call the catarrhal stage, which is pretty much like having a bad cough and a cold, snuffles. That lasts for a couple of weeks. And then it progresses to something much more horrible, the paroxysmal stage, which will go on for 2 to 6 weeks, or sometimes even longer, where you have this horrible racking cough, paroxysms of this cough, which can really be very distressing both for the patient and for those caring for them. And then there's a long convalescent stage where the patient is exhausted from having suffered the infection and takes quite a long time, months perhaps, before they get back to full strength again. And there are these so-called sequelae, things that follow on. We get weight loss, something called surgical edema. So the coughing can be so bad that you get air blown into bits of the tissue, say around the neck and into the areas around the lungs, and you can get brain damage as well. How does this disease come about? Well, there are a whole clutch of different virulence factors associated with pertussis. There are a number of adhesins, Two in particular named there, the filamentous hemagglutinin, which binds to a particular sulfatide residue on the cilia of epithelial cells, and then something called patactin, which is a protein that lives in the outer membrane of the bacterium, and it allows them to stick to the tracheal epithelial cell. So it's in the trachea that we see this organism proliferating. There is also what we call a type 3 secretion system in this organism. This is a kind of molecular syringe which allows the bacterium to inject effector proteins into host cells. And later in the course, in coming years, if you do more microbiology, you hear more about this. There are these toxins as well, the so-called Pertussis toxin. Again, this is an AB toxin. It's actually an AB5 toxin because it's got one active subunit, which is called S1, and then there's this ring of 5 binding subunits, 2 of which are the same, and then 3 which are different. And again, this is an ADP ribosol transferase, but this time it has a completely different target. It attacks heterotrimeric G proteins and interferes with cell signaling and causes damage to cells that way. Similarly, we've got this adenylate cyclase toxin also, which binds to calmodulin, and then you get this unregulated production of cyclic AMP from ATP. How do we diagnose this? Well, if we look in the blood, we see something called lymphocytosis, a proliferation of lymphocytes in the peripheral blood. So when you're take a blood cell and you look at the white cell count, that's one of the hallmarks. You also can grow the organism, and it normally lives at the back of the throat. Particularly, the best place to try and get the organism is from the back of the nose, the so-called nasopharynx. What you do here is you actually pass a swab through the nose, a particular very thin, a swab on a very thin piece of wire that you can thread in through the nose and get right to the back of the nose and sample there. Treatment is the same as with diphtheria. In the catarrhal stage, you can give erythromycin, but by the time they actually get symptomatic with that horrible whooping cough, it's too late. The disease has acquired its own momentum and just keeps going, and there you just have to give them supportive treatment. Like with diphtheria, though, the antibiotics do prevent infectivity. And again, there's vaccination available with this acellular vaccine as part of the routine childhood vaccination. Just worth briefly digressing into a kind of public health perspective here. It's clear that vaccination does work. If you look at the graph at the top there, when immunisation was brought in the 1950s, you saw the number of cases plummet dramatically. But then what happened, we had several decades, a couple of decades of good, effective use of the vaccine. And then in the 1970s, there were these unsubstantiated claims of a link between the vaccine and brain damage. It's just one of these media scares that got a certain momentum, no scientific basis to it. But because of this scare, people stopped vaccinating their children, having their children vaccinated, and we saw the disease come back again. In the last 10 years, we've also seen a dramatic increase in the USA, and it's unclear why Pertussis is resurgent again. But both of these graphs should warn you not to be complacent in the battle against infectious diseases. We have many tools that get rid of infection from society, but these infections do come back if we let down our guard. Right, let's now move to the gut and talk about this organism, Helicobacter pylori. So this is a gram-negative bacterium. It's highly motile, it's spiral-shaped, and it zigzags around, swimming around with this, these flagella that you can see here at one end of the bacterium in the electron micrograph. It causes inflammation of the gastric mucosa, gastritis. It's also associated with duodenal ulcers and gastric cancers, gastric cancers as well. It colonises the front part, the antrum of the stomach, and it colonises the epithelium there. We don't quite know how it gets around. It seems to be spread in families, kind of by direct person to person, maybe kissing, that kind of stuff. We're not really certain. But one of the strange things in epidemiological terms is this is an organism that's on the way out. Now, some people would argue it's a commensal, because if you go back in time, then perhaps the majority of the population carried this organism in their gut, in their stomach. If you look at people over the age of 60, around 50% of them are infected. But if we go and look at people under the age of 40, they're only a minority, around 20% are infected. And we're seeing it disappear across the globe. Now, this is quite an interesting phenomenon and quite a controversial one. There is one individual, Martin Blazer, who says that actually it's it is a commensal, but it's a good commensal because it prevents us from getting carcinoma of the esophagus by tuning down acid production in the stomach, and that getting rid of it is not necessarily a good idea. But that's his opinion. That's not the consensus at the moment. Now, the interesting thing about H. pylori is the story of its discovery. It's one of these things that there are lots of prequels, if you like, to its discovery. It was observed many times from the 19th century onwards, but never really followed up. And the conventional thinking was that the acid bath of the stomach is just such a hostile environment that nothing could actually live there and make that its home. But in the late 1970s, Australian pathologist Rob Warren, shown here on the left, was working in Perth, and he had seen the organism down the microscope. We'd never been able to grow it, but he started working with this physician here, Barry Marshall, in 1981. They couldn't grow it. They tried and they tried until one Easter weekend, they left their plates up over the whole weekend for 5 days instead of just leaving them overnight, which is what they were doing before. And that was enough to actually allow them to grow the organism. So they then published in The Lancet, the premier journal of medical science, and there was a lot of people poo-pooing it. They weren't really clear. Is this a real thing? If these bacteria do live there, are they really associated with peptic ulceration like these people are saying? Is it that they're just colonising abnormal stomach, diseased stomach, but they're not actually causing the disease? So to demonstrate that H. pylori caused gastritis, not merely a bystander, Marshall then in 1985 went on and drank a beaker full of H. pylori culture. He took some bicarbonate of soda and I think he mixed it up in a kind of milkshake and then swallowed it down. And he developed a very bad case of nausea and vomiting. And then when he was endoscoped, it confirmed that there was gastritis when there hadn't been gastritis before, and that H. pylori had colonised his gastric mucosa. You could argue that he was a bit mad, but you know, this was a very cogent demonstration of the fact that it was actually a pathogen. Subsequent to that, antibiotics are shown to be effective in the treatment of gastritis and the prevention of the relapse of duodenal ulcers. So back in the late 70s, early 80s, we started to have effective treatments for duodenal ulcers with things that suppress acid production for the first time. But whenever you stop those, the thing came back. But now it was shown that if you take away H. pylori, you prevent that relapse. And in effect, Warren and Marshall rewrote the textbooks with the reference to what causes gastritis and peptic ulcers. And when I started out, as an undergraduate back in the late 70s, what you see at the top there in those pictures, this was some kind of hemigastrectomy. These are the sort of horrible operations that people would do in extremis to try and cope with recurrent duodenal ulcers. They cut part of the stomach out and part of the small intestine. Horrible operations. And all of that's been replaced now by simple, cheap antibiotics that do the same job, in fact, do the job better. And as a result of these efforts, Warren and Marshall were awarded the Nobel Prize in 2005. One of the most important virulence factors of H. pylori is urease, which neutralizes acid. It breaks down urea, produces ammonia, It's a way in which the organism can assimilate nitrogen sources through the ammonia, but the ammonia that's released also damages the mucosa. And you can see there one of my proudest accomplishments early in my career as part of the team that actually first cloned and sequenced the urease genes from H. pylori. More recently, the protein's been purified, the structure's been solved, which you can see there. There are a range of other virulence factors associated with H. pylori in addition to urease. We need the flagellum, this kind of whip-like organelle that gives it motility. There are a number of exotoxins and enzymes. And there's a different kind of molecular syringe here known as a type 4 secretion system, which injects a particular effector, the CAG-A effector, into host cells. And this has all sorts of effects on the cytoskeleton, actin remodeling. And when you look at the cells down the microscope, these are the epithelial cells, they take on what's known as the hummingbird phenotype. Rather fanciful term, but with the eye of faith, you can look at a cell and say it looks a bit like a hummingbird. Okay, we're nearly finished now. Last infection I wanted to cover in this lecture was cholera. So this, it's not in the stomach here, we're now looking at the large bowel, and we have Vibrio cholerae, a curved gram-negative rod, as the causative agent of this infection. There is one major serogroup worldwide, so-called serogroup 1, which is responsible for human cholera. You do get other Vibrio cholerae strains which have not yet been associated with human infection. Then within that, there are these two kinds of so-called biotypes, the classical and the L-Tor biotypes are being described. There's also, in the late 80s, early 90s, we saw the rise of a completely different serogroup, the Bengal 0137 serogroup, named after the Bay of Bengal, where it was first described as causing infection, but then subsequently found around the world. Now, cholera is transmitted by contaminated food, or more likely, or commonly, contaminated water. And it has these clinical features of producing this profuse watery diarrhea. The stools from a patient with cholera are said to resemble rice water, the kind of stuff you get if you boil some rice for your dinner and then you look in what's left in the saucepan and you've taken the rice out. That's pretty much what it looks like. This is a cholera hospital here, I think in Bangladesh, and you can see that what is required to nurse these patients is a plastic lining there on the bed and then a hole through which the stools can pass into a bucket underneath, because these individuals produce up to 10 to 20 litres of diarrhea a day. And the major challenge with cholera is just keeping up with that fluid loss. And it's that fluid loss that kills the patient. Nowadays, with intravenous fluids, we can keep up. But in days gone by, that kind of loss of water was just enough to kill you. We can culture this organism on a particular selective medium known as TCBS, but you don't really need to know that small print stuff. Now, Julia Lodge tells me that you know all about this here. Have you heard about this particular thing in the picture there? Yeah? So you heard about John Snow and his observations in London during a cholera epidemic, that the majority of the cases clustered around a particular pump, and he took the handle off the pump and that signalled the end of the outbreak. And in fact, for those of you who've got a very keen eye, if you can see that pump there, which has been put there as a memorial, it's not the original pump at all. You can see in the background there is a pub. That pub is actually called the John Snow Pub. There's a John Snow Society which you can join. The sole requirement for joining that society, the sole condition of joining it, is that whenever you go to London, you go to that pub to have a drink. We are racing against time, but we have a few minutes left. The epidemiology of cholera is interesting, and sadly this disease has not gone away. And a couple of years ago there was this outbreak in cholera, and to bring things up to the modern age, genome sequencing actually established that cholera was brought to Haiti, not from traffic with local islands in the Caribbean or South America. Instead, it arrived with the UN peacekeepers who'd come from Nepal. And tracking down that source of the infection actually had quite severe political ramifications. Cholera, there's this adhesin, the so-called toxin co-regulated pilus, but instead of mediating adhesion purely to the mucosa, this actually tethers the bacterial cells together, so they form this kind of bundle, and they resist the shearing forces that are trying to force them out of the intestine. Cholera toxin is encoded again on a phage, so again this is a kind of similar motif that we see in pathogenesis in various different contexts. This is an AB5 toxin again, with a B subunit binding gangliosides, and then this A subunit, again another ADP ribosyl transferase, this time targeting G proteins. And what happens is you get a derangement of the physiology, you get cyclic A and B produced constitutively, and this leads to secretion of water, sodium, potassium, chloride ions, bicarbonate ions into the lumen of the small intestine, causing diarrhea. So that's me finished. I have managed to outrun the battery and survive. So that's us finished for today. We'll talk about some invasive infections later in the week, and I'm happy to take any questions now.